Dr. Kulkarni. Now, this is the third period of enhancement in food production. Here, I have mentioned on the board some heads: micropropagation, disease-free plants, and the production of secondary metabolites, as well as somatic hybridization. These four are here the applications of what we are saying tissue culture. These are of these four, micropropagation is the first one. In this micropropagation, what we are doing? We are preparing number of propagules in a very short period of time, in a short space, and season independent. Here, what we are doing in this activity, this is a type of tissue culture practice. In this case, generally we are preferring to take the stem apex, and that apex is allowed to grow, stem apex as an explant, and allowed to grow in the nutrient medium. Here, the same apex is allowed here to form number of apices. That can be done by changing the hormonal concentrations. Normally, we have mentioned when cytokinin is applied, cytokinin is high in amount compared with the auxin, kaulogenesis takes place. If the cytokinin concentration when exceeds, the kaulogenesis takes place, number of plantlets will get formed. All such plantlets are further here removed out from the explant, and each one is now ready to form a new plant. All are the clones because all are dividing mitotically. All the cells in the apex are dividing mitotically, and all are the clones here, and such in a very short span of time in the laboratory, with that one, the seasonal independent number of plantlets one can generate. This is called as micropropagation. Second one is tissue culture technique is also come into rescue with disease-free plants. How disease-free plants are being produced? Tissue culture technique we have studied in the earlier period, we have gone through it. In this tissue culture technique, what we are mentioning, the tissues are taken out, these are aseptically cultured in the nutrient medium, and later after development of organs, here that one is hardened and planted in the field. Hardening is done and planted in the field. In production of disease-free plants, what we observe normally, to the apical area, normally virus or virus-like pathogens are not infecting, neither virus nor others, because that area, apical meristem area, is highly active area, and on that area the viruses and other pathogens are not infecting. That area remains free. From the diseased plant, one can collect the healthy plants by removing the apex of the diseased plant. That apex of that diseased plant is further cultured in the laboratory and later on produces the healthy plants. This can be done in the plants which are what we say, we are saying plants which are with the good quality, with the desired types of the characters. Such a desired type of character containing plants can be recovered by the process of tissue culture. This is what we are calling the disease-free plant tissue culture. Likewise, Here, with the help of the cell suspension culture, we have mentioned the the cell suspension culture in the last period. In the cell suspension culture is a culture where the liquid culture is there, and in that liquid culture the cells concentration is high in amount, and after each 7 to 8 days the same culture is being is freshly supplied, because the growth of the cells is high here, and no cells are involved in binding with each other. They remain free. Such as cells of the noted or the noted plants are being utilized here for what? For production of the secondary metabolites. Here various metabolites we have mentioned on the board. Here the vincristine, vinblastine is there. Likewise anthocyanin is also there. Tropane is there. Menthol is there. And nicotine is there. All these different, what we are seeing, secondary metabolites, these metabolites are separately harvested from the plants here. Vincristine and vinblastine are the anti-cancer drugs, are collected from Catharanthus roseus. Likewise, anthocyanin is an alkaloid collected from Dauthus carota. Datura stomium is involved in giving us a secondary metabolite, propane. Menthol, you all knows, menthol plant is involved in providing us the menthol alkaloid, secondary metabolite. Likewise, nicotine is harvested from the tobacco plant. These are, here the plant, their cells are involved in here production of secondary metabolites. Tissue culture, with the help of tissue culture technique. And the last one is, one can mention, somatic hybridization. In this somatic hybridization, what we are mentioning, two vegetative cells are being hybridized with each other. Here in that hybridization technique, say for example, you knows very well, one plant cell is with cell wall, this is nucleus, cell is here. Another cell, one can say from this is the cell wall, this is the cell wall, this is nucleus, cell and its inclusions. That cell wall is removed out, and here the plasma is here, protolemma is here, and nucleus is here, cell wall is removed out. This is what we are calling a cell with protoplast. No cell wall is here. Such a protoplast containing cell is now is fused with a fusogenic agent called as PEG. PEG, for the PEG is what we are saying is polyethylene glycol. With the help of this fusogenic agent, such two cells which are without the cell wall can combine here, and a cell may get generated that contains both the nuclei A as well as nuclei B, containing what the protoplasm is. Now, this one is with the characteristics of two separate plants, and what they produces a combination of two plant here, and because of this somaclonal hybridization we can produce different plants. Here an example is there. Pomato, we know pomato is a combination of potato and the tomato. Likewise, Raphano brassica is what we are seeing. Raphano brassica, One cell protoplast is of Raphanus and another cell protoplast is Brassica. Such two protoplasts are fused with fusogenic agent PEG, and the single plant can produce two products. In earlier case, the potato and the tomato. In latter case, the Brassica, the mustard, and the what we are calling the cabbage. This is what all are the applications of the tissue culture here. One more point is left here, and that one is SCP. SCP stands for single cell protein. What single cell protein is? Actually, this is here the biomass, dried biomass, and that dried biomass is utilized as food as well as feed. The question is why? Because here the population of humans as well as animals is increasing fastly, and to that pace the food production through the agricultural department is not producing. Because of this reason, here number of peoples are suffering from malnutrition as well as here the hunger. Because of hunger and malnutrition of the protein content as well as vitamins, minerals, here because of this reason a huge demand is increasing, and that demand cannot complete by the agricultural department. That's why an alternative way to generate food as well as feed is developed here, is produced here. That one is here what we are calling single cell protein. Single cell proteins are generated from microorganisms, single celled microorganisms or multi-celled microorganisms. Single celled microorganisms from the field of the algae, from the field of the bacteria, from the field of blue-green algae. Here examples I could quote on the board is single-celled alga is green alga is chlorella. Second one is here what we are saying, the spirulina is a blue-green algae. Blue-green algae. And the third one is what we are calling a member from the bacteria is Methylophilus. Methylotropous. Here, these members are utilized in producing the dried cell biomass. That dried cell biomass is used as food as well as feed for the animals. You may note here, not only this biomass is acting as a food, but this also contains minerals, vitamins, and that can satisfy the things needed to the poor as well as the hidden hunger peoples. One can observe an interesting point about this Methylophilus methylotropous, and that one is when Methylophilus methylotropous is allowed to grow, that can produce a huge biomass. Before knowing this one, where these microorganisms can grow, they can grow on such substances. For example, spirulina, one can say that can grow on the waste material from the potato industry, as well as on the molasses, as well as on the manures of the cattle. On these material that can grow here, and with this one without forming any pollution-free growth can takes place in the laboratory or in the area, and in that area a huge amount of the protein containing food is developed. Normally what we observe here, a 250 kg cow can produce 200 grams of protein per day. But it is noted, interesting to note here, when Methylophyllus methylotropus is of 250 grams is allowed to produce cell biomass, dried cell biomass that can produce in a single day 25 tons of the single dried cell biomass can be prepared, can be produced by the Methylophyllus methylotropus when compared with a cow. Cow can produce only 200 grams of protein containing protein here in a day, and takes how much? It takes 3 to 10 kg of the grains, 3 to 10 kg of grains. Further that reload on the food grains, which is already meager and cannot satisfy the growing population. Whereas that can produce a very huge in amount and becomes a food as well as fit to the cows as well as the chickens. This is what the enhancement in food production. Here we will stop. This topic is now concluded here. A new topic we will start, and that topic is called as biochemistry of the cell. Thank you.